start? I'm very sorry we don't have more room. We didn't anticipate quite so many people would be here. We will try to find larger rooms next time. Please all keep this a secret from the fire marshal, and you'll be okay, at least for today. Very glad to see you all. I hesitate to say it, but if you're not already on our mailing list, go to evmedcenter.org to get on the mailing list to figure out what's coming. And also, I have a special request. Please RSVP so we know how many of you are coming, so that we can adjust food and size of rooms appropriately. So today's a special treat. Joe Alcock is someone I was eager to have come give a talk, because there are only a handful of actual practicing physicians in the United States of America who really know evolutionary biology and are trying to apply it in medicine. It's also a special treat, and he's one of them. Also, I think you came to me first 14 years ago, something about then?
Longer than that.
Longer than that?
Yeah.
I'll pretend it's 14 years. And he came to the University of Michigan applying for the MD PhD program because he wanted to get a PhD in evolutionary biology and get his MD at the same time. And I said, that's really great. This is exactly what people should be doing. And we went to the committee and they interviewed him and said, you're a brilliant scholar, but we won't allow you to get a PhD in evolutionary biology. That's not relevant for medicine. Since then, actually, they've had me twice to the MD PhD program in Michigan to give their annual big deal lecture and convince people about how interesting it all is. So that's fixed now, but it was too early for him. He went on after his undergraduate degree at University of California, Santa Barbara, where he was biology with high honors, to Cornell, where he got a master's degree and did study neurobiology and doing work in the field as well. He went from Cornell with a master's degree to get his MD at UCLA. And for the last several years, he's been at the University of New Mexico, where he's the chief of emergency medicine, and also running an evolutionary medicine course. So he's kind of half-time, kind of the same kind of career path I've taken, where he's half-time in the College of Liberal Science and Arts and teaching, and half-time running a very active medical practice. It's a very rare thing. He also, somehow, in the midst of all of that, is managing to do research and publish. Most recently, an article with Carl O'Malley and Adina Actipis about how gut microbiomes might, in fact, manipulate us to eat things that are good for them, which he'll tell you about. And I don't usually offer editorial comments ahead of time, but when I first saw this article, I was worried that some of my favorite people were going way, way out on a limb. And when I read the article finally, though, they have some really good specific tests about how to actually test these ideas. And I'm so glad you're here today to tell us about it.
Fantastic. Well, thank you, Randy. And it's a great honor for me to be here, and I appreciate the invitation and the opportunity to speak to all of you. I've definitely been inspired by Randy Nessi's writings and our communications over the years. And like Randy, I think that it's impossible to do medicine and to practice as a physician without some understanding of evolutionary biology. It's inconceivable to me that there are people out there that don't have a background in evolutionary biology. It's been hugely helpful in my own career, and I think in terms of my ability to take care of patients, it's enriched my own life and some of my research ideas. And none of that probably would have happened without the book that Randy Nessi wrote with George C. Wallace back in the day. So thanks. And it's also a special treat for me because ASU was a stomping ground for me when I was a kid, having grown up in Tempe. And by the last time I was on this part of campus, I was probably skateboarding around as a teenager. So it's fun to come back in a different context. So today we're going to talk about, I think, things that many of us are interested in, which is sort of why it is that we might get fat, and what drives our eating behavior. So, what makes us want to eat the cookie or the sandwich or an extra serving of potato chips? And I think that something we're thinking about as you're digesting today. So, I was asked to come up with a kind of a title that would be of interest, maybe kind of catchy. So, we're at Allies or Enemies, Gut Microbiota and the War on Fat. So, an alternative title might have been the title of our recent article, which, let's see if this works. Here we go. Is eating behavior manipulated by the gastrointestinal microbiota? Evolutionary Pressures and Potential Mechanisms, and my co-authors are in the room, Athena Actipis and Carla Maley, who contributed more so than I did, probably in terms of bringing these ideas to fruition. But it was definitely a joint effort and an important one. So when you're looking at me, it's important to think that I'm not just a mammal. I am a meta-organism. All of you and all of me, we're outnumbered in terms of mammalian cells and microbial cells. It's a 10-to-1 ratio. So there are 10 microbial cells to every human cell, and the numbers get worse if we think about the number of genes in our bodies. So of the 100 trillion microbes that are mostly in our lower intestine, they contribute a huge number of genes to us, what we think of as being ourselves. So if an alien came down and took a gene at random from us, they would be much more likely to pick up something from a gut microbe than the mammalian cell. So a Martian may conclude that in fact we are more microbial than human. And this has really changed how we think about lots of things in terms of human health, in terms of how evolution, about how we interact with other organisms, and I think important for this talk about the potential for some of these interactions to affect our behavior. We are apt to think of ourselves as autonomous, of making up our decisions about our choices that we make throughout the day and having some sort of free will. But it may be, in fact, that some of our behavior, some of the things that we do, some of our food cravings, for instance, might in fact represent another genetic interest. And to give you some background about this, people have written about coevolution of humans or mammals and microbiota. And there's a literature that goes back at least a decade, because the microbiota field is still relatively new. But this is a paper written in Nature in 2007 by Relman and his group. And they make the argument, in effect, that the relationship between us and our microbiota is one of a mutualism, that we have evolved with to mutual benefit. And in fact, he argues, as encapsulated by this quote, that the individualized microbiota of each person has a stake in his or her fitness, which would suggest that we are co-evolved. A symbiosis is one in which, characterized by mutualism, in which each partner gains something from the other and enhances the fitness of the other. This may be true, but it certainly isn't the only outcome of how two genetically different organisms can interact. And it's equally possible that we could be parasitized by some of our gut microbiota, and that the fitness may actually be reduced. That these fitness interactions may not be so simple as mutualism. Having said that, probably some of the most influential people that have written and talked about the microbiome, so for instance, Marty Blaser of NYU, makes the point that, in fact, many health problems result from the absence of microbiota among modern humans. And probably people in the audience, you've heard this argument before. I think it's true. I think that, in fact, we know that autoimmune diseases, allergic problems, allergies, those of you that suffer from seasonal allergies, that these things don't happen in other populations that have a more microbiota-rich environment. And in fact, the whole idea of the hygiene hypothesis relates to this idea that we are missing some old friends among our microbiota. They've been lost along the way. Antibiotics, exposures, too clean of an environment, that these things have actually led to outbreaks of disease. But this, again, reinforces the idea that there's something good about our microbiota, which I don't disagree with, but it's not the only view. Our problems do not relate just to the fact that we're missing a microbe or two. It could be the relationship between mammal and microbe is far more complicated than that. Another meme that you see in the literature is this idea of the microbiota as a forgotten organ. And this phrase, forgotten organ, or has been cited multiple times from dating to about 10 years ago. And the idea is thus: it's that the gut microbes serve a bunch of functions. In fact, they do. They generate vitamins like vitamin K. And they do some things that our bodies don't do particularly well, or not at all. But the argument that the microbiota is an organ, like the heart or the lungs or the eyes, I think is specious, because in fact we can live without the microbiota. And we don't know this from people, but we know this from other animals, that we can have germ-free animals, and there may be germ-free facilities at ASU for all I know, in which animals are housed in a completely sterile environment, and those animals don't just die. They don't keel over dead. In fact, they can live quite happily, and in fact, some of the early studies looking at germ-free mice suggest that the germ-free mice live longer than their conventionalized counterparts. You can't think of too many other organs that do that, where you can't take out the heart and have you live longer. Maybe gonads, but there's some special cases, right? So I would argue that this idea of a neglected or forgotten organ, there's some, because of the genetic differences between the microbiota and us, they don't behave in the way that, say, one of our organs does. So let's talk a little bit about germ-free animals, and we're going to relate this to obesity, because you may be skeptical about the idea that gut microbes are responsible for weight gain and obesity. But this has been demonstrated pretty nicely with the series of germ-free animals. So you can so for one thing, germ-free animals are resistant to obesity. A study done by Frederick Backhead in the mid-2000s showed that mice that were genetically predisposed to be fat remained lean if they were raised in a germ-free environment. So the sterile animals stayed skinny. As soon as they were inoculated with a microbiota from another mouse, they restored the gut microbiota and they rapidly ballooned up. This made it look like obesity is like an infectious disease, that it can be transferred like a cold or flu or Ebola, for that matter, from one individual to another. And it really kind of changed how we think about obesity, not just being a matter of calories in, calories out, but the fact there's another genetic interest in there that's modulating our fat storage. What's kind of cool is that if you take microbiota from an obese mouse and give it to a germ-free mouse, that mouse will get even fatter than it otherwise would. If you take microbiota from an underweight mouse and inoculate it to a germ-free mouse, these mice actually can lose weight. And if you take microbiota from a normal weight mouse and inoculate it to a germ-free mouse, the weight doesn't change. So these studies have given pretty good proof of principle that microbiota are not only, there's not just an association between microbiota and fatness, but in fact, there's a causal relationship. This suggests this is true. Now, to the extent to which our fatness in humans relates to our microbiota, it remains a bit of an unsolved mystery. Clearly, we're not mice, but our physiology is somewhat the same, and we might expect that the microbes are behaving in a similar fashion. So this, again, in this instance here, fat microbes causing fat mice would suggest the microbiota are not always acting in the best interest of their host. And I'll give you one more example. I just found this from a PubMed search a couple days ago, just published, showing again that there's some advantages to being germ-free. If you're wounded, mice that are germ-free, they heal up faster, and they have wounds that heal up without any scarring. So the fact that we have scarring seems to be somehow related to whether we have microbes. So being germ-free is sometimes a good thing, and might actually improve our fitness if we lacked gut microbiota, perhaps. Something to think about. The only message here is that our relationship is more complicated than either being a pure mutualism or a pure parasitism. It's probably somewhere in between. So listen, the idea that we are kind of a complex holobiont, or that we have a metagenome, that mammals plus microbes, has some interesting implications for evolution. And these ideas are inspired in part by the work of David Haig and Bernie Crespi and others, who did a work suggesting there can be interfamiliar, intergenomic, intersomatic conflict that is within the same organism. So probably the most famous example would be during pregnancy. So we know that pregnant mothers share about, on average, 50% of her genes with the fetus, and the other 50% on average would come from the father. It turns out that there are some maternal fetal conflicts that relate to a variety of diseases like gestational diabetes and pregnancy-induced hypertension that are an outcome of this conflict. And again, the theoretical framework from this relies on work done by Trivers and others. From a medical perspective, what we see is that there are imprinted genes that are responsible for the growth of the fetus, and the genes that are important in the insulin-like growth factor and the receptor for the insulin growth factor are imprinted. They get turned on or turned off depending on which parent you inherit them from. And it suggests that the very slight difference between the fetus and the mother can have a big impact on a disease or not. And the fact that this is a intersomatic conflict between how resources are used and how much resources gets delivered to the infant. I could spend all day talking about this, but I'm not. But just to let you know that we would expect that even with small differences in relatedness, you might expect some genetic conflicts. So how much are we different from our microbiota? If we're 50% the same as between mother and fetus, if you think about us versus our gut microbes, we're 0% related between us. Do you guys know where this image comes from?
Voyager.
It's a Voyager. So it's out there in space somewhere beyond our solar system. It's on a gold etching that's meant to tell aliens who discover this something about the creatures that produced it. No microbes in this picture, by the way. But this is E. coli, or one of our intestinal microbes. We don't share any genes with the bugs in our guts. That would suggest that there's an opportunity for conflict, just like we see with the maternal-fetal conflict. Changing gears slightly, so what's the likelihood or the possibility that having an organism that lives in us could change behavior? It turns out that there's some good evidence from the animal kingdom that a parasite can radically influence behavior. And this is a nice example. This is the cordyceps fungus. It turns out there's a very nice YouTube on this if you just put it to Google that's worth watching. It turns out the cordyceps fungus can infect a whole variety of different insects, and it radically changes the behavior. Most notably, this is an ant that's been infected. In general, ants living in the forest stay on the forest floor in this species, but when they're infected with the fungus, the ant will leave the colony and then climbs the very highest plant that it has access to, and instead of staying on the forest floor, it will go up several feet. At that point, the fruiting body of the fungus emerges from the brain and from the rest of the ant's body, and it turns out that the change in the behavior, that the hijacking of the neural machinery by the fungus of the ant helps the spores of the fungus find new potential ants to infect and improves the fitness of the ant. So clearly, here's an example whereby an organism that lives in the same body is reducing the fitness of the ant, but promoting the fitness of the microbe. So it's worth thinking about. Gosh, well, these are ants. This is kind of a bizarre phenomenon. Zombie ants. Does this happen in people? Gosh, who knows? And really, the jury is out. So I'm not by no means am I suggesting this is a case closed phenomenon in humans. But let's talk about mammals. The classic example for parasites manipulating mammalian behavior involves toxoplasmosis. And this is an example whereby these mice that were infected with toxoplasmosis, which is a protozoan, a parasite, they lose their aversion to cat urine. And in fact, they may actually be turned on by it. They get sexually aroused in the presence of cat urine, and they seek it out. So this is great for the parasite because the mice then get gobbled up by cats. And then the life cycle involves a cat and then the prey species of the cat. So the parasite changes the behavior of mice. Uninfected mice show a normal and healthy, for the mouse, aversion to cat urine. So at least for toxoplasmosis, it looks like, if you're a mouse, we can see some of this dynamic of genetic conflict between the symbiont and the host. Could this be happening with gut microbiota? What we do know is that diet has a big impact on who's there and what they're doing in our guts. If you eat a plant-based diet versus an animal-based diet, it causes rapid and big shifts in the gut microbiota. The implication is that diet selects for which are the winners and losers in our guts. And we know this can happen relatively rapidly. There was a study done last year by Peter Turnbaugh and his colleagues, and he showed that here we're looking at some metabolites that are produced by the bacteria, but we could just as easily show you a slide that shows that the actual composition undergoes massive shifts over a short amount of time, whether one eats a vegetable-based or an animal-based diet. And this is true for virtually any kind of nutrient shifts that we see these kinds of changes. So in other words, you could imagine that the microbes in our guts have an interest in what we eat, because either they have a selective advantage on a certain diet or they have a disadvantage. So that's based on sort of which nutrients we consume. You can make a similar argument for how much nutrients we consume. So the overall amount of energy that we intake has a big impact. And fasting changes the composition of microbiota versus overeating. So both the quality and quantity of our diet selects for microbiota. From an evolutionary point of view, you might suppose that if the microbes have the capacity to manipulate behavior, as they do in cordyceps or in toxoplasmosis for mice, that the microbes might have an interest in getting us to eat their preferred nutrient in the quantity that optimizes their fitness. So that is the underlying hypothesis that we suggest in the paper. There's some evidence from other species that this might happen. The microbes have a potential for benefit. We know that certainly diet affects human fitness and in ways that may be different than for microbes, and that has the potential for conflict. But I gave you a couple examples of fungus, a eukaryote. I gave you an example of protozoan. How about a single-celled organism? Can a bacterium affect behavior? It can, again, if you're a mouse. And we know this is also probably true for people. This is a cool study done a year or so ago, in 2011, in which the investigators discovered that the NIH Swiss mice were different from the Balb C mice in terms of how anxious they were or how anxious they behaved. The way you measure anxiety in a mouse in one way is to see whether it explores its environment. And so the latency, how quickly it will jump off a platform and go exploring would indicate a non-anxious mouse that is brave. Well, these investigators found that if you get mice from a certain supplier with a genetic makeup, you have a brave mouse, and the brave mice are the NIH Swiss mice, and the Balb C mice are more anxious and timid, and they don't go exploring their environment. These investigators wanted to know how much of these behavioral changes are dependent on microbiota. They raised the mice germ-free, and they inoculated them with microbes from the other kind of mouse. And they found that you could switch the phenotype. You make a brave mouse timid. You make a timid mouse more brave. And these are things that are there's a causal relationship. The nice thing about the germ-free models, you can determine causality. And we found that you can change the behavior depending on how they're inoculated. You can switch it around. So pretty cool, pretty amazing. Suggests that, in fact, some of the things that we take, if we can extrapolate mouse anxiety to human anxiety, things that we think of as personality traits that are intrinsic to ourselves may, in fact, be modulated, at least in part, by our microbiota. So again, we've not clinched it. We've not shown that bacteria can affect food choices. Is there any example of bacteria affecting nutrient delivery affecting the host in a way that affects their nutrient delivery? There is, although this may be a little bit obscure to some of you. It turns out that if you take a microbe at random from our guts, they fall into one of two phyla, the Firmicutes or the Bacteroidetes. So this is Bacteroides theta iota omicron. And it manipulates or modulates activity of the host in a way that favors its growth in the gut. It does this by, instead of changing the eating behavior of its host, this is a grazer that grazes on mucus in your gut. And it stimulates host epithelial cells to produce glycans, complex polysaccharides that they can then cleave and consume as an energy source. And it's been shown quite nicely that the activity of the microbe induces the host to produce the nutrient substrate for its growth. And there are other bacteria that do the opposite, that actually will reduce the glycan production by the host. So the one that doesn't is this Faecalibacterium. Interestingly, so the sugaring, or the glycans that are expressed in the intestinal epithelium, this happens also when you get sick. So if you get really sick, and we can do this with a lipopolysaccharide challenge, you inject lipopolysaccharide, all of a sudden your intestinal cells become sugared, encouraging the growth of this commensal bacterium. So interesting dynamics going on here, but it's a proof of principle that bacteria can control, under certain circumstances, their nutrient delivery. We don't know yet whether they can control our behavior in ways that change our eating behaviors. But let me give you one potential example. This is Colic. Have you guys seen this? So the photographer is Jill Greenberg. Has anybody seen these photos? So people have a whole series of these images where the infants are crying. They look like they're having a terrible day. And she's been accused of child abuse and worse. But it turns out the way she does this is that the mothers willingly bring their babies in to be photographed, and she hands the baby a of candy or a sweet of some sort, and then she takes it away. And this is the moment after she takes it away. So the kid is not being physically tortured in any way. Just so you know. Yeah. And I include this. So, of course, kids with colic cannot be consoled. They always look like this. And it's extremely upsetting to parents. Parents bring, I've been in the emergency department where parents will bring their children in to be evaluated for colic, and we look for various things. We poke them and prod them, and most of the time we find nothing wrong with the child. But it can be very distressing for the parent, and parents get less sleep. They're definitely stressed. They're anxious. David Haig has some ideas about how that could be adaptive. But colic doesn't seem to be bad for babies of interest. And so people have speculated that, in fact, infant crying has a signal function that elicits support and, in fact, feeding from its parent in ways that may be beneficial for the baby. So the important thing to know about colic is that colic is not harmful. Babies that are colicky are typically born a little bit heavier, and they have more weight gain after a month than babies that never have colic. So fatter, heavier babies tend to do better from a whole variety of different health measures. And it doesn't appear to be harmful. More harmful for the parents, but not for the baby. So the hypothesis that some have suggested with the social origins of infantile colic is that perhaps colic has some benefit in terms of eliciting additional feeding for the baby. But clearly the infant doesn't look happy. They're crying, they're upset. And so others have wondered if maybe there's another genetic interest in colic and have studied whether microbiota might be different in colic. And in fact, there's some good evidence that it is. This is one figure from a recent study in 2013 in which the signature of the intestinal microbiota looked different in infants with colic versus without. The main difference that we're highlighting here is that colicky babies have a less diverse microbiome. So there's fewer species present. The complexity of the intestinal ecology is less rich than babies that don't have colic. And that'll be interesting. So we, in our paper, speculate, and again, this is just a speculation, that perhaps microbes are inducing some changes in the baby's behavior that allow the parents to feed the babies and deliver more energy to them. So just keep that in mind. And if this is true, if we think about mechanisms by which this might happen, and again, this is highly speculative, the mechanisms might involve making the babies feel worse. So the baby actually is experiencing something, pain or distress or anxiety, but the outcome is that the baby gets fed more. So this may be a potential example whereby microbes could influence behavior that relates to energy delivery to the infant. And in fact, a couple of subsequent papers have shown that you can change this in colicky babies and give probiotics, change the overall diversity of the microbiome, and reduce some of these colicky symptoms. So perhaps the word's not out yet on colic, whether this actually happens or not, but it's a testable thing that's worth investigating. If it is true that microbiota are somehow producing substances that could influence the brain or behavior of a human, how might this happen? This has been worked out in some great detail by a researcher by the name of Mark Light, who's currently at the University of Texas. And he has shown over many years that there's a nice relationship between bacteria and human neurotransmitters. And in fact, bacteria produce the exact neurotransmitter that we rely on for brain function, behavior, and mood. And I'll give you some examples. So milk-associated bacteria like lactobacillus and bifidobacteria, they produce GABA. The GABA receptor is the same one which is activated by Valium, so it makes you calm and relaxed. Pathogens like E. coli or Bacillus species, they produce norepinephrine. I'm not talking about something which resembles GABA or norepinephrine. I'm talking about the exact same molecule. They produce this stuff. And norepinephrine, instead of making you calm and relaxed, of course, it jazzes you up and it gets you excited. Serotonin, the chemical which is commonly thought to be important in mood, along with norepinephrine and dopamine, is produced by a whole variety of different microbes. And the dopamine neurochemical, which is involved in reward pathways and other things, is produced by a variety of microbes as well. So these are commonly found in our guts. In fact, we all have these inhabitants in us, and they produce neurochemicals. Could these neurochemicals be escaping from the gut, going into the bloodstream, and going to our brain and affecting us? There's some evidence that that is true. On the other hand, we produce chemicals and enzymes to degrade these so monoamine oxidase and other enzymes that remove them from basically the milieu, to prevent them from entering the bloodstream. So that indicates to me the potential for genetic conflict. Microbes are producing a neurochemical that has the capacity to influence behavior. The host is producing an enzyme which degrades that neurotransmitter. This may be an example of a genetic conflict between host and microbiota. This slide suggests that there's a whole variety of different ways in which microbes might affect us. So the neurotransmitters, the dopamine, serotonin in the gut is one, but it turns out that microbes produce toxins and virulence factors that might influence eating. Microbes also alter the expression of taste receptors in our mouth and in our gut. We know that there are opioid and cannabinoid receptors in our intestines that are modulated by microbiota. There's a wide variety of different ways in which microbiota might potentially adjust our behavior. And again, these are potential pathways, potential mechanisms for a phenomenon that's not been all that well worked out, at least for people. One example that I find interesting as well is that in addition to neurotransmitters, bacteria produce peptides, hormones. They produce peptides that have a great degree of homology with human hormones and, in fact, act on the same receptors. I just highlighted a couple here, orexin and alpha melanocyte stimulating hormone. These are both really important in stimulating appetite, and there are others up here that do the same thing. So these are peptides produced by bacteria that make us hungrier. Why are bacteria producing these things? It turns out that there's some environmental microbes also produce these peptides, so we can't argue that the only reason they're doing this is to manipulate us. In fact, they may have evolved sooner or earlier in microbes before they evolved in humans. We may have co-opted some of these, the signal functions of these peptides, perhaps from microbes. But the bottom line here is that they produce things that make us eat more, in line with some of the expectations of this hypothesis. And with regard to the potential conflict or the push-pull between host and microbe, humans produce antibodies against these bacterially derived peptides. So that would suggest that we're trying to reduce any kind of interference of signaling that bacteria are producing. Of note, in terms of the effect on human health, in anorexia nervosa, there are autoantibodies against orexin and alpha melanocyte stimulating hormones that also attack the human peptide and make people with anorexia nervosa not want to eat, which, again, for someone like me who's interested in microbiota, I'm saying, wow, check it out. Microbiota are involved in everything. It may be true. But for things like anorexia nervosa, so we think of it as being a purely psychological disease, it may be that microbes are somehow important in eliciting an autoimmune response that is important in that disease. So I'm going to give you a couple of slides that relate to some work that I've done, and then hopefully we'll wrap things up. People that have sleep apnea produce a huge amount of norepinephrine, and I found a linear relationship in a study that I've done in human subjects between the abundance of gram-negative bacteria and norepinephrine that those microbes might be either producing or exposed to. This is an association, but again shows that in people there's some relationship between neurotransmitters and microbes that are important in health. We've been doing some bench research looking at hydrogen sulfide, which has a neurotransmitter signaling function. And these are mice given sulfate-reducing bacteria. They produce this neurotransmitter gas in their guts that makes the mice kind of forgetful and stupid, and they can't remember where they've been. So there are some human disease states in which sulfate-reducing bacteria are important as well. And so I include this slide just to show that there are multiple potential pathways by which microbes could influence brain and behavior of a mammal. So if this is all true, and if microbes are involved in obesity by some change in behavior, then we should be able to do something about this because we can manipulate microbiota. We can do this with antibiotics, we can do it with probiotics, we can do it with fecal transplants. Has everybody here heard of fecal transplants? So it's gotten a lot of press recently. It's exactly what it sounds like. So in this study, they fed mice a fast food diet, things with simple carbohydrates, low fiber, and high saturated fat. Lo and behold, the mice became fat. They gave a lactobacillus probiotic, and they protected these mice from obesity. This is one of a variety of studies using different kinds of probiotics, suggesting that maybe we can manipulate obesity, and perhaps in part by eating behavior, using probiotics. I'm going to give you a slide from a different study that shows that in this one they used a different probiotic, one called VSL3. And the animals that were fed a high-fat diet that would otherwise become obese and consume more calories, these animals were protected and ate less when they were also given a probiotic. So we know that by manipulating microbes in the gut, we can change the calorie consumption of the animal. And so perhaps this is something which is modulated in some way by microbiota. Now, if you remember, when I was talking about colic, I mentioned that the colicky babies had a reduced diversity of the microbiome. People that are obese tend to also have reduced diversity of the microbiome. And we were just talking a moment ago about bariatric surgery, which of course is a surgical treatment for obesity. People that undergo bariatric surgery, they lose weight and they are less interested in calorie-dense foods, and they change their eating behavior, and the diversity of the gut goes up. So there seems to be a nice relationship between the diversity of the microbiome and eating behavior and obesity. So let's talk a little bit about diversity. If you've been following the argument so far, I've been suggesting that perhaps microbes can produce substances like neurotransmitters or other peptides that influence host behavior. The inference from this is that if this is true, we'd expect that if there was a dominant microbe that overgrew its habitat and was existing in greater abundance, it would have a greater capacity to influence the host via these molecular mediators. One could imagine that if we have a highly diverse microbiome with lots of different microbes, each producing different signals, that no one group would have be able to attain a population density great enough to affect the host. And so host signaling would be unimpaired. So the receptor and the signal production by the host wouldn't be influenced by the microbiota. But imagine if you have an overgrowth situation in which a single group dominates and produces a large amount of dopamine or norepinephrine or orexin, the appetite hormone. It's possible there could be some interference between host signaling. So one testable hypothesis from our paper that we wrote is that the diversity should be important in any kind of manipulation between host and microbiome, and that perhaps we could reduce the potential for manipulation by affecting diversity. The thing I'm going to close with is to tell you that everything I've said so far, we're drawing these connections between microbes and obesity and various things. There was a paper just published by Rob Knight's group about the microbiome, trying to decide, well, which are the microbes that are the worst? Which are the ones that are causing the obesity? And they did a meta-analysis of a variety of different studies. And the really disappointing thing was he found the studies were conflicting and they were contradicting each other. In some studies, Bacteroidetes were the ones that were responsible for weight gain. Others, it was firmicutes. Some were proteobacteria. And we've not been able to pin the blame on any particular microbe. It looks like the activity of the microbiota is more important than who's there. And one thing that may be regulating gene expression among microbes that's important for obesity and behavior and health in general might be diversity. So we know from other studies that low diversity seems to be associated with bad health outcomes. So now I'm kind of getting back into the idea that perhaps having a diverse microbiome is good for us. We know that low diversity of the gut microbiome occurs in Europe and North America. It's higher among, say, hunter-gatherers like the Ache. It's higher among Malawians that have been studied, and also in Japan. We know that diversity is low in urban populations, higher in farm living or rural populations. We know that diversity of the microbiome is low. If you eat a low-fiber McDonald's heavy diet, it's higher if you have a high-fiber Mediterranean diet. We know that obesity is a low diversity state, being lean is a high diversity state, and even hospitalized patients have a low diversity. So patients that I see in the hospital, we give them antibiotics, we wipe out the diversity. We know that hospitalized patients have low versus community. So one perhaps objective for health might be to encourage having a high diversity microbiome. One of the reasons why this may be beneficial might be that it interferes with the capacity for any individual microbe to manipulate its host. So are they allies or enemies? It's a complex microbiological world. We don't yet know whether our cravings for Whataburger or a milkshake is determined by microbiota, but there are ways to get at this question and test these hypotheses. One of the things would be to actually do the transfer of microbiota between humans the same way it's been done in mice, and find out whether some of these things occur in people. Perhaps probiotics are the answer. We're trying to figure out which ones are the most beneficial for health. Those things are not yet known, and whether they affect cravings is another question entirely. Alternative hypotheses for things like food cravings and obesity seem to be falling flat on their face. So the idea, for instance, that we crave things that we have a nutrient scarcity of just does not match with evidence. The idea that all of obesity is simply a matter of willpower or a matter of calories in, calories out doesn't seem to match with the evidence. So the idea that the microbiota is somehow involved is better supported by alternative hypotheses for obesity and for some things about cravings, at least that I've proposed here, but the final answers are yet to be tested and explained. So I think with that, I will take questions. Yes?
So, is there a difference in the rate of colic for formula cards versus breastfed babies?
I think so. I'd have to go back and
repeat the question, please.
So the question is, for colic, is there a difference between the colicky behavior in breastfed and formula-fed babies? I think I've read that, but I don't have a reference to put my tongue.
And then do you know, so I thought that I heard recently that diversity was lower in the microbiome in formula-fed babies versus breastfed babies.
So I believe that is true.
Okay.
So breast milk is pretty amazing stuff. Over 30% of the carbohydrate fraction of breast milk is complex carbohydrates, oligosaccharides. These human milk oligosaccharides cannot be absorbed by the baby, so the mother's producing an energy-rich substrate that the baby can't absorb. What does absorb the energy? Microbes. It's a fermentation substrate for microbes. And it looks to me, and there's some people like Katie Hind who have been doing work showing that the composition of the milk oligosaccharides might be designed in such a way that they encourage the growth of bacteria differently for different populations. So cool stuff. We predict that these human milk oligosaccharides for breastfed babies would encourage a diverse microbiome versus formula-fed that don't have these oligosaccharides might be less diverse.
Cesarean babies, do they have a less diverse biome?
So I can say that more confidently. So babies that are not born vaginally acquire a different microbiota, one that is less complex, one that has more pathogens. And that has big implications for things like asthma, HOP, and autoimmune disease. Babies that are born by C-section have a higher risk of those kinds of problems. Yeah?
Can you maybe speculate about the pathways by which microbes could trigger specific cravings associated with specific kind of foods?
Well, so we had a slide that showed the multiple different pathways. There's different ways. I can't give you an example because this is we're suggesting something that requires additional investigation. One pathway is that we know that, for instance, we have inhabitants of our guts that are pathogens. Things like E. coli, things like Serratia, things like Helicobacter pylori that Marty Blazer thinks is not a pathogen. So we know that these things they commonly inhabit the gut, but don't cause us any harm. It turns out that the virulence factor expression of some of those microbes is determined by what nutrients they're exposed to. So if we give them sugar, for instance, they don't produce the virulence factor. And this has been well established in vitro, that virulence factor expression from a whole variety of microbes can be inhibited if you give them a simple carbohydrate solution. They don't produce it. Why change lifestyle and do something which is risky, which is to turn on virulence programs and attack the host when they have free, easy food? So one of the ways in which manipulation might occur is by virulence factor production. So we speculate about that in our paper. Let's see. Yes?
So in cases where you get bacteria producing these signaling molecules like GABA, acetylcholine, dopamine, where do those genes come from in the bacteria? Is it possible they're picking them up from the host?
Sure, it's possible.
Can you repeat the question?
The question is, so neurotransmitters, so the neuropeptides like acetylcholine, dopamine, serotonin, GABA, etc., that we know that bacteria, various kinds, have the genes that give them the metabolic capacity to produce these neuropeptides. Is it possible that we might have gotten those genes from bacteria? Is that the speculation?
Or vice versa.
Or vice versa.
The bacteria get them from the host, because they're just such weird things for a bacterium to produce.
Right. So I can tell you that even environmental microbes do produce these. So maybe they're producing peptides and neuropeptides for reasons that have nothing to do with mammals. That it modulates some interaction between them and competitor species or quorum sensing or in free-living, non-human associated bacteria. I don't know the answer to your question. Mark Light, who has done much of the work on this, he speculates that these neurotransmitters evolved first in bacteria and that we mammals co-opted them in some way. Yes?
So if I understand correct, the diet that isn't diverse would kind of influence that one microbe could become dominant, and that could lead to manipulation.
That would be a testable hypothesis. So
Okay.
perhaps does dietary diversity affect microbial diversity? Pretty good evidence that's true, especially if you're eating whole foods, things with complex plant polyphenols and complex carbohydrates. That seems to encourage diversity of the microbiome. So does dietary diversity then affect cravings? We'd expect that it would reduce them. So we could test that idea.
So the idea of the competition between the different microbes, the idea that if it's sort of balanced and it's diverse, none's getting above the threshold to actually be able to have the influence on the. So there's another metaphor one could use for conflict. I mean, this is just reproductive, reproducing, and maybe they're just competing for the resources. But do these microbes, they actually attack each other? I mean, are they aggressive in these kinds of ways and control each other those ways as well, or just be a competition for the available food sources?
So the question is, instead of just simply, so we can model this and we can imagine that bacteria populations are going up and down depending on whether they have their growth substrate present as a pure energetic sort of phenomenon. The question is, are they attacking each other and doing various things? So I mentioned, if we go back here, hydrogen sulfide. Hydrogen sulfide produced by the sulfide-reducing bacteria is highly toxic to other bacteria. It does some very interesting things. Hydrogen sulfide has a neurotransmitter effect in people and us. It's also produced by these bacteria, but it kills off their competitors. It's directly toxic to them. It does another thing, which is it affects the enteric nervous system in a way that instead of having peristalsis, where things are moving through the gut, things stop. So it gives this bacteria a specialized niche in the gut and allows it to persist in a place that it otherwise wouldn't be able to. So I think that it provides a good example of competition with the host for ecological space, and the fact that it kills its competitors is exactly what you were saying. Let's see, bring your shirt back.
Moving beyond obesity, how does this relate to things like inflammatory bowel diseases like Crohn's or ulcerative colitis, things like that? Is there any kind of relationship there between microbiomes and severity of symptoms, things along those lines?
So there's probably better evidence for things like for inflammatory bowel disease. The question is, how does this hypothesis relate to diseases like inflammatory bowel disease? There's better evidence for IBD, that microbiota are involved, than for many other diseases, even obesity. We can actually find signatures of specific microbes that seem to confer an additional risk of IBD, whether that's causal or associated, we don't yet know. I will tell you that for things like Crohn's disease and ulcerative colitis, which are the two archetypal inflammatory bowel diseases, that both of those are associated with a decreased diversity. So it kind of goes along with some of these ideas. I don't know if anybody knows whether in IBD people have specific food cravings. So, did you have a question?
Yeah. So I was thinking that if the bacteria are winning the conflict between the host and the bacteria, shouldn't the host not be getting fat? I mean, the fact that the host is getting fat implies that bacteria are not winning the conflict.
Good point. Really, that's an excellent question. So the question is, the fact that when people are getting obese would suggest that the host is the one who's able to monopolize the resources, and it's not going to bacterial growth. And it was suggested that if there's a conflict here, the host is winning, and that the microbes are losing out. I think that's a really an exceptional point. Presumably, in the case of obesity, still more energy is being consumed, And I would argue that the guts have sort of prioritized access to that energy. They can get to it first, even before we can. And in fact, some of the obesogenic things that microbes produce are some of the fermentation products. So they go through the microbe first, they produce short-chain fatty acids. Fatty acids get taken up in colonic and enterocytes. That seems to lead to obesity. So there's some complex dynamics here. But they're definitely getting access to the food first.
But the fact that in the more diverse populations you get skinnier hosts might suggest that you have increased virulence or something when there's more competition between strains of bacteria. And so the more homogeneous bacterial populations are being more cooperative, maybe.
Also a very interesting point. So perhaps more virulence factor, perhaps the opposite of what we suggested here is that in more diverse communities, we see more virulence factor production. I imagine there might be some instances in which that's the case. It's a testable hypothesis.
Joe, this gentleman back here has
sorry.
his hand up for a
That's
long time.
I was just curious about your choice of word for your title. These guys have been with us for a long, long time, right? So from an evolutionary perspective, why would they be our enemies? And there would be an evolutionary truce at worst, but
I think it's a sort of marriage of convenience. I think you're right. So for instance, one of the first citations that I put up, the paper by, I'm blanking on it now. It was the Rollins group, suggesting that there's a mutualism that's co-evolved. He argues that exact point, that we humans, sort of like the island biogeography of humans as being a habitat for these microbes, that we can trace some phylogenetic changes over time and between different organism groups of microbiota. And that, yeah, if they rely on us being alive, why would they ever want us to die? Because, so that gets at questions of sort of the evolution of virulence. And what I can tell you is that very clearly, our bodies contain potential opportunistic pathogens. They're just waiting for their opportunity to turn on virulence programs. They turn on us and they do kill us. We know this is true.
how often is this true in hunter-gatherer kind of populations where the diversity is much higher?
So maybe all the badness that we see in terms of virulence, maybe inflammatory bowel disease, obesity. Maybe this is also the fact that we have a messed up microbiota that's different from hunter-gatherers. There are people. Leach was in science. He actually gave himself a fecal transplant from a Hadza hunter-gatherer. You guys read about this? Pretty amazing stuff. No, without screening for parasites or pathogens. So there are people that are interested in this question. And he suspects that he may undergo some beneficial physiological changes as a result of transfusing himself with...
He lived with a Maasai or something for a while.
It's Hadza. It sounds like, yeah. So yeah, people are actively investigating that question.
Just to sort of follow up on the question about fat and whether being fatter is an indication of the the host winning. Actually, you're the one who sort of talked to me about how microbes can influence the gut permeability and how omental fat is different from other types of fat. So I wonder if that might be a piece of this, and also just the fact that having more fat isn't necessarily a good thing for fitness, because the whole the body has to sort of balance the needs to get around with how much fat is being stored. And so I was hoping that maybe you could speak to that a little bit.
I will.
Okay.
So again, getting back to your question, which is that does the fatness seem to indicate the host is winning, or so an alternative hypothesis, which I did not discuss, although I'm very interested in, and we've discussed this, is that what's the evolutionary function of fat? Is it purely manipulation? Probably not. We have people that have some adiposity do seem to have benefits in certain disease states. Certain starvation, it's better to be fat. And one thing that seems to happen is that if you do have bacteria leaking from the gut into the bloodstream, your body responds by increasingly depositing peritoneal fat or omental fat. And that fat can wrap intestines, and it does this in appendicitis. That if you have an appendicitis or omental fat, then basically encases the appendix and keeps the bacteria from spreading the rest of your body. That's a very concrete example of how fat can protect you from dying. And it suggests that fat, under certain circumstances, has a host defense function, and that fat may, with relation to the gut microbiota, one of its functions may be to contain it to the gut where it belongs and keep it from invading the rest of the body. So there's some complex alternative hypotheses that are worth exploring. So the potential to which this whole system can be manipulated by microbiota is one question. The other question, which hasn't been discussed too much in the literature, is why do we have fat? Why does the capacity to have gut fat in particular? Why do we humans have that capacity? And is it adaptive? Unknown yet. If that's what's going on. Carlo.
Yeah, just to get back to the other conversation, that just the fact of a long evolutionary relationship does not imply beneficial mutualism, right? There are diseases, there are pathogens, there's parasites that have ancient evolutionary.
So tuberculosis and cholera are 2 examples of human association. Certain long evolutionary association between mammal and microbe does not always lead to mutualism. And that, I think, is a settled question in biology.
I was just wondering if any research has been done on the microbiota and cravings in substance users.
I don't think anything's known, but we've talked about this a little bit, which is the idea. So alcoholics clearly crave alcohol, and we know that alcoholics undergo massive changes of their microbiome, because many of them don't consume trace nutrients and have a very reduced diet, and many alcoholics get alcohol or energy from alcohol that transforms the microbiome. So we have thought about whether it's possible that certain microbes might induce cravings for alcohol, for instance. Interesting question, and I think one that needs some kind of follow-up. But just also, those neurotransmitters, people that have used methamphetamines, that is related to norepinephrine, the opioid receptors that are important in regulating gut homeostasis, the fact that we're injecting vast amounts of that or taking pills, clearly these things have effects on the microbes that you could imagine some kind of a feedback loop. But then for those kinds of things to have evolved, you'd have to speculate that those are environments that have been around long enough for the bacteria to evolve that adaptation. That's unclear.
Let's have one more question, and then we'll wrap up.
Sure.
So I remember reading recently, or fairly recently, that rabies in kind of the later stages, in addition to things like lockjaw, can also cause like hydrophobia and aversion to water. Has kind of the link of that process and the microbiota process been kind of compared or looked at all?
So the question is, in rabies, the fact that it involves, causes encephalitis and changes the behavior of people that are infected, and one of the changes of behavior is hydrophobia. People that have the later stages of rabies cannot drink water; they froth at the mouth and drool. The question would be, how does that relate to this process? We could include that as an example of a parasite or pathogen changing the behavior of its host. It's probably the best example in people of this happening. And it's very rarely described in terms of its adaptive value to the virus. But clearly, animals that are frothing at the mouth and biting other creatures, that happens in humans less so than perhaps in predators, enhances the fitness of the virus.
I have a brief announcement, but before I go to that, let's thank Joe for winning.
Thank you guys.